Okay, we're wrapping up the Protozoa chapter. These little projections from Flagellates and Ciliates. One thing about them, interestingly enough, their internal cores are identical. They have this little ring of what I call microtubules around a core of a couple of microtubules in a 9+2 arrangement. And even though these tend to be very different sizes, they're built the same way. And I don't believe the microtubules are different sizes. I don't actually know that now that I'm thinking of it. But here's your either or list. Flagella tend to be bigger, cilia tend to be smaller. Common flagella are way bigger than common cilia. There's an overlap in between. There's no like one threshold where if it's this above the threshold, it's flagella, and below the threshold, it's cilia. There are some pretty big cilia and some pretty small flagella. And it's other stuff on the list that kind of breaks the tie. But typically, flagella are big and cilia are small. When you find a cell that's got flagella on it, the odds of you finding a cell that's got more than a dozen flagella on the cell are very, very, very small. There's just a handful of cells that would have 13 and up. There's a weird group of flagellates that live in wood-eating insects, actually digest the wood for them, that they look like they're covered with long hair. But they're weird, and so far as I know, that's pretty much it. Whereas when you look at it, ciliates, even if they're not covered in cilia, which is fairly common, they have bands of them that will be hundreds and hundreds and hundreds of cilia. So the numbers are way high versus 12 or less. It's quite a distinction. Not a big surprise. When you've got a whole bunch of cilia packed together and they're trying to produce a wave of motion across the cell so the cell can swim, or you can move something past a barrier, there's ciliated barriers in a lot of multi-celled animals, then they gotta work together. You don't want them all working in different directions. They get tangled on one another. Whatever movement you pull across the surface it's all gonna be very turbulent and very chaotic. They need to work together. Whereas when you have multiple flagella on a cell, keep in mind that this screen that I'm looking at is backwards, so sometimes I gesture at the wrong thing. Sorry, but the words are backwards as far as what I can say. Anyway, flagella, even when there's a few of them, they work together but kind of loosely. I'll put the list down, and then I'll give you a little story about something that happened to me that was a very unusual thing to have happen. Since flagella are bigger, they can be modified in various ways. They can have combs on the end of them or attached to a membrane like a fin, or in some cases have a stiff part and a wiggly part. Is that there's a lot of ways you can modify them with these little add-on structures. Cilia are much smaller. They tend to work as groups. The closest you get to an add-on structure is some cilia will actually fuse together and produce a structure that's kind of a combination of a bunch of cilia, but that's not really an add-on to a single cilium, so that doesn't really work. They don't really have add-on structures. So here's my story. There's a very common flagellate that lives in ponds that would have a little nucleus inside it. And it's got two flagella. And the flagella are stiff for about a third of their length, and then they're wiggly for about 2/3 of the length. So they swim along like a boat with two outboard motors on it. And I'm looking at pond water one day in the lab, and one of these guys goes swimming by, and before he gets across the field, one of the flagella falls off. I've talked to people who work on these things. They go, what do you mean the flagella fell off? So this is an unusual thing apparently to happen, that a bunch of people I've talked to who observe these things go, I've never seen that happen. But it did. The flagellum came loose, it wiggled for a little while, it kind of eventually drifted off of the field I was looking at. But with only one of these things working, because the wiggly thing is at an angle, this thing comes out stiff and then it's at an angle, is that with only one of the things working, it couldn't compensate, and all it did was spin. Which was great, because I didn't really have to chase it to keep watching it. But over the course of about 15 or 20 minutes, it gradually grew another flagellum where the first one had been. Now, it did it differently than I would have expected. I expected the wiggly thing to grow first, and then eventually a stiff part would push out of the cell. But what it actually grew was the stiff part, so it kept spinning. And then when the stiff part hit the same length as the stiff part in the other one, a little wiggly end started to progress out. And the farther out it got, the less it spun and the more it spiraled, because now it was starting to get pushed from both of the flagella. So, not real coordination so much as long as they're both working, the cell's okay. And eventually it grew to its normal size and it swam off, and I eventually lost track of it. But that is a type of add-on where you've got not the entire flagellum working the same way. And that's just a weird thing to have happen. One more thing, we were talking about ciliates, and I want to mention that ciliates do sex in a very bizarre way. So what makes us us? It's all of our stuff. It's all of our potential. And you can make a case for your potential being embedded in your mixture of genes, your mixture of alleles. That if we were to come in and completely, fiddle with all of your alleles, you wouldn't be you anymore. That's what happens when ciliates have sex. Two ciliates will swim along and they meet each other. Now they can reproduce asexually too. They just make new copies of micronuclei and divide, distribute them. But these guys do what's called conjugation. They get next to one another and a bridge forms between the two participants. It's not gendered because they're both doing the same thing. There's no different roles here. What happens in each cell is the micronucleus, which, as happens in eukaryotes, has two sets of everything, two sets of chromosomes. It does a division where it'll make a little special micronucleus with only one set. So it kind of sorts through all the pairs and gets a combination. The other one does the same thing. It sorts through all of its pairs and gets a combination, and then they trade. And the one that goes across fuses with one of the other ones that was made by mixing and matching the pairs. And the same thing happens over here. And those are going to be the new micronuclei when those things separate. So when those things separate, they're not genetically the same critters that made contact in the first place. They now have a completely different, not completely different, they have pretty different mix of genes. So it's like you'd have 6, and when you were done, you were a different person. That's what happens with these guys. And then they almost immediately do asexual reproduction. They make copies of this new mixture to kind of perpetuate it. But it's a very weird setup where it's essentially sex is suicide. If the end of yourself is essentially your suicide. And they seem to be very happy with it. Okay, one more group of protozoans. This is an example of a group that got its name changed. So you pick up an old book, these will have been called the sporozoans, the spore animals. And that's a sucky name. Spores. Bacteria produce spores, plants produce spores, funguses produce spores, protozoans produce spores. Is that the people that worked on them said, spore animals is just a lousy name. It's too broad, it's too generic. We want to rename it to something specific to this group. And one of the things about these cells is they have a little complex of stuff near, the cells are asymmetrical. They have kind of a little pointy tip called the apex. And so they have what's called an apical complex. And they figured, who cares about us guys anyway? We make this long, nasty name. We know what it means. So they're now called apicomplexans. This is a group that is purely parasitic. They purely exist living in other living things, usually inside bigger cells. This is another group that does alternations of generations, which is very common in parasites. It's almost like if you're a parasite, getting for your offspring onto another potential host, the right kind of host, that's a pretty daunting challenge. And remember, both types of reproduction have their own advantages. You can make copies of successful forms, and you can combine the genetics of successful forms. And that's what these guys do. But they do it in different hosts. They'll do their asexual form, make copies in the intermediate host, and then they'll do sexual reproduction, which they do in a very weird way, a lot of them, where they kind of change, where one cell takes on kind of the male characteristic where it becomes a swimmy seeker cell, and the other one will build up resources for the offspring cells, so it becomes kind of the female cell. But that happens in what's called the definitive host. And these two terms will show up in other parasites that also do this same sort of approach. Give you an example. There's a lot of examples of these guys, but one of them you've all heard of. Actually used to happen around here, malaria. Malaria actually shut down the building of the Erie Canal for a while during a particularly nasty hot summer out near Syracuse. They had a huge outbreak of malaria and they couldn't continue until things cooled down and mosquitoes went away. Now, the mosquitoes for malaria are the definitive hosts. That's where sexual reproduction happens. We, and other animals that malaria can live in, birds mostly, are intermediate hosts. A mosquito lands on you and it needs to suck some blood out of one of your blood vessels. So it sticks this long needle-like nose into your skin, and then this long wiggly thing comes out the end of the nose that's pointy at the end, and it looks to find a blood vessel, and it pokes the blood vessel. And it's gonna suck blood up that little wiggly thing and the long nose. And when you suck blood through a very thin tube, it will immediately clot. So they need to keep the blood from clotting. They have an anti-clotting agent in their saliva. We're talking about the malaria. They spit down the tube, and then they can suck blood through it. The malaria, after it's done its reproductive cycle, embeds itself in the salivary glands of the mosquito. So when it spits down the tube, it spits malaria right into the blood of the new host, if it's a person, into us. And so these parasites, they don't stay in the circulation long. It was a big mystery for a while because they would disappear and then reappear later. Well, it turns out as soon as their circulation takes them through your liver, they invade your liver cells. And they will sit in there and reproduce for quite a while. But they don't do a lot of damage. Your liver cells are quite big. They're kind of tough. And the reproductive cycles doesn't seem to damage the liver cells. And if it did, you regenerate liver cells pretty quickly anyway. In some people, that's it. Because your immune system has gotten exposed to them and is building up antibodies to these guys. And some of them, your immune system is so good that they can't emerge back from your liver before your immune system kind of wipes them out, kind of kicks them down really fast, and you don't get anything. But for most people, what emerges from your liver is a wave of parasites. And they're not done. They invade red blood cells and start to reproduce in there. Remember sickle cell anemia? They get in there and they make more malaria. Your immune system goes berserk. It's trying to kill them. It's trying to go after the infected blood cells. It's trying to kill them with heat death. You run such a high fever that it cooks your brain. The symptoms of malaria is basically your immune system kills you. Is that it's trying so hard to kill the malaria, it actually kills you doing it. Unless it doesn't. But now the malaria is out circulating in your blood, so the next mosquito can pick it up. And the cycle continues in the next mosquito. So that's your typical malaria. Another apicomplexan that you could get exposed to around here is Toxoplasma. Toxoplasma is, its actual preferred intermediate host is cats. And its definitive host are fleas. And it lives in cat digestive systems. The eggs pass in cat droppings. Fleas lay eggs, they drop off of the cat, and the eggs hatch, and they form these little larvae that crawl around and just eat things, including these eggs. So that's how the flea larvae get reinfected with the eggs, well, the cysts, they're not eggs, the cysts of Toxoplasma. And now the larval forms stay in the fleas, reproduce, and eventually the fleas transform into adult fleas, get on a cat. At some point, a cat licking its fur, biting at an itch, swallows the flea. They do that a lot. And when you digest the flea away from the Toxoplasma, it now infects the next cat. People can host Toxoplasma. Doesn't really like to live in us. It doesn't do a very good job of it. Most people exposed to it don't even generate antibodies to it because it just is really bad at it. But in theory, it can invade people and it can embed itself in your nervous system. They have found that people very, very immunocompromised sometimes have a Toxoplasma eruption from their brains, which can be bad. People with regular immune systems, it just never gets a chance to do that. It also can cross into a developing embryo when the early in the embryo, when the embryo is first developing a nervous system, it can mess that up. But that's pretty unusual. But there was a time where, pregnant women were told to get rid of their cats. It was not really a risk, but people went nuts. You actually don't commonly get exposed to Toxoplasma from cats anymore. It also can get into other animals that also will lick at their hair and pick up fleas and stuff. Cows and sheep. So in the United States, Toxoplasma can sometimes be embedded in meat, in beef, and you can pick it up, and pork. And you can pick it up if the meat's not cooked fully. If it's bloody beef it's unusual, pork it's more common. They used to not let you sell rare pork because there's a worm we'll talk about later that was a problem that they pretty much have under control. But at some point when they decided to allow pink pork to be sold, they kind of forgot about toxoplasmosis. And food poisoning sources of toxoplasmosis is the most common way that people get Toxoplasma now. So even though legally you're allowed to serve pink pork, if your pork comes in it's pink, send it back and have them finish cooking it. On top of everything else, I always do this in restaurants. I drive my wife crazy. I go, no, it's pink, you gotta take it back. There's something in the meat that could be dangerous. And the waitress goes, oh no, we get our pork from local farms. And as you'll find out later when we talk about the worms, that's not good for the worms. You're much more likely to get worm exposure from a local farm than these huge factory farms. And that's a mystery for another day. I will tell you about that later when we talk about roundworms, my favorite types of worms. But those are the protozoans. This wasn't too short a video tacked on to the end, but just to let you know, you're not the only one getting tired of hearing me talk. I'm getting tired of hearing me talk, doing all these videos.